Hello, my name is Ed Chapman, and this video cast is going to cover the processes of cell division. Now, prokaryote cells are our simplest cells in biology, and they are otherwise known as bacteria. And because they're simple, they divide very simply in a process called binary fission. Now, binary fission just splits one large bacterial cell into two smaller but identical bacterial cells. And in order to accomplish this, all the cell has to do is grow larger, copy its loop of DNA, here represented by this purple stringy material, separate the two copies of DNA, and as the DNA is separated, simply pinch itself in half by cleaving right here in the middle. So at the end of the process, you end up with two identical daughter cells, process known as binary fission. Now, binary fission can divide cells pretty quickly. For example, some of the E. coli that can grow in your mouth may grow as quickly as one cycle per 20 minutes. So you can end up with a mouthful of millions of bacteria about 8 hours after you brushed and used mouthwash. So that's why you have to always repeat that process. Now, eukaryote cells are more complicated. Eukaryote cells, such as plants, animals, fungi, algae, and protozoans, their cells possess nuclei. And because they have a nucleus plus cytoplasm, they have to go through some extra steps or phases to divide their cells. A little bit more complicated because of their cell structure. Now, eukaryotes divide their nucleus first, and then they divide everything else, their cytoplasm, which includes all their organelles. They do this in a life cycle known as the cell cycle. And it's called a cell cycle because it moves in a circle. So most of the time, this is represented as a sort of a clock-shaped diagram here with arrows moving in a clockwise direction. Now we're going to start right here, and if we go all the way around once, through one complete cell cycle, we will have turned an original cell into two identical daughter cells. And in order to get from here all the way back around to there, we have to go through interphase, and then something called mitosis, which is divided up into 4 parts. We're going to talk about each of these parts in more detail. So interphase is the first part of the cell cycle. And if you remember, right there. All right. Interphase is the first part of the cell cycle, and inter means between cell divisions. And for a long time, scientists, when they observed living cells, couldn't really tell what was happening during interphase, so they didn't think much was going on because it wasn't visible. All they could see is that the cell wasn't dividing yet, so that's why they called it interphase, which means the phase between cell divisions. Most of the cells in your body right now are in interphase. They're just living and doing their jobs until they wear out or until they die. They will never actually divide. In this picture, you can see we have I think this is a fish egg or a fish zygote. Those are often used to take these types of pictures. You'll notice in here the nucleus is darker than the surrounding cytoplasm. This dark material in here was named chromatin, which literally means dark stuff in Latin. And over here to the right, you can just barely make out a little fuzzy darker area. This is a centriole, and centrioles are involved in cell division. But before a cell can divide, it has to copy its centrioles. So in this picture, you can see that they haven't divided yet. That's just one set of centrioles. Now, in interphase, if a cell receives the signal that it needs to divide, then what it's going to do is it's going to grow to maximum size, and it's going to replicate its DNA. It's going to make a copy of all of its chromosomes. And once its chromosomes have been duplicated, then that cell is obligated to divide its nucleus. It has to complete the cell cycle. Now, the first part of mitosis is called prophase. And a cell that's in prophase, or the first or pro part of mitosis, a cell that's in prophase, its chromatin, that dark granular material we saw earlier, is going to contract and condense into visible bodies called chromosomes. And the word chromosome in Greek means dark bodies. So in this picture you can see here that we've experienced a change here. In this cell, the chromatin has started to condense into these wormy looking structures. All these cells around it, they're not doing that. So all of these cells around the cell that the arrow is pointing to are an interphase, but this cell here has started to do something different, and we're going to call that prophase. Now, each of the chromosomes inside of here, remember, because the DNA has been duplicated, each of the chromosomes is actually made up of 2 sister chromatids. Now, what in the world is a chromatid? So let's explore that in a little bit more detail. if you think of chromatin as a bowl of kind of yellowish spaghetti material here, and you wrap this yellowish spaghetti stuff up around these meatball kind of shaped things called histone proteins, you end up with a condensed body called a chromosome, which is actually made up of the spaghetti, which is DNA, and the meatballs, which are the histone proteins. So a condensed chromosome is actually about 50% DNA and 50% protein. And the point where the two copies are stuck together is called the centromere. Now let's look at that a little bit more close up. So here's the centromere, and here we have one chromatid over here and the other copy over here. So let me draw a ring around it. So inside of this loop is one sister chromatid, and across from it is the other sister chromatid. So together, this is one chromosome, but remember, each chromosome is made up of two identical sister chromatids still attached here at the centromere. Now, prophase is where the nuclear membrane is going to break down and vanish. The centrioles are going to replicate and begin moving apart. And prophase is going to be complete when the nuclear membrane is gone and the chromosomes have completely condensed. So in this picture here, we can see the centrioles, here represented by these little asterisk-shaped things. The centrioles have copied, and the two copies are moving away from each other. And we also have in here 4 chromosomes. In this particular picture, we have 4 chromosomes. 1, 2, 3, 4, and you can see each chromosome is actually made up of 2 sister chromatids held together at a centromere. The next step of mitosis is known as metaphase, which means middle. And in this case, the centrioles, which have moved as far away from each other as possible, start to spin out a spindle apparatus here made of microtubules. And as these microtubules lengthen, they're going to be attaching to the chromosomes. And as the chromosomes have spindle fibers attached on both sides, they're going to be pushed and pulled to the middle or equator of the cell. So we call the area where the chromosomes are being lined up the equator, and the 2 areas at opposite ends of the cell are called the poles, and the poles are where the centrioles are. And as you can see here, the spindle fibers are bracing and pulling and pushing to elongate the cell into kind of like an egg shape or an oval and line the chromosomes up in the middle. Metaphase is finished when the chromosomes are all nice and lined up here in the middle. Now the next stage of mitosis is called anaphase. And anaphase is where the sister chromatids are going to be pulled away from each other. And this happens as the spindle fibers get shorter. So remember, each chromosome, this red chromatid here and this red chromatid here, which were once connected as a single chromosome, but the spindle fibers attached to each side have pulled them apart. So now, once they're completely separated, we refer to this as a chromosome and this as a chromosome. So the word chromatid is only a temporary word. It's only really used when we have 2 identical copies of the same chromosome stuck together after replication. So once one chromatid moves this way and the other one moves this way, we enter anaphase. Now anaphase sometimes can experience mutations. Okay, here's a really common example. You probably remember Down syndrome, also known as trisomy 21. So if we look at this cell here, this chromosome was supposed to have been pulled apart with one chromatid going this way and one chromatid going this way, like we see down here. But that didn't happen. For some reason, this spindle fiber didn't attach. And because there was no attachment, this whole chromosome got pulled this way. Both copies went that way. So we're going to create a cell down here which has an extra chromosome. And of course, the one on the other side is going to end up missing a chromosome. This is one type of mutation that you're probably most familiar with if you remember Down syndrome back when we looked at karyotypes. The last stage of mitosis is called telophase, and this is the end of mitosis. And what happens during telophase is each pile of chromosomes, which are now at the poles of the cell, will become new nuclei. And this happens when new nuclear membrane is built around them. So in this picture, remember, these chromosomes have moved, and now you can see represented here by this dotted line, a new nuclear membrane is being formed around this pile of chromosomes to produce what's going to become a new nucleus. And the same thing is happening down here at the other end. Now, while this is happening with the DNA or the chromosomes, the whole cell's cytoplasm is being separated here in a process called cytokinesis. And in this picture, these arrows represent kind of a pinching in of the cell membrane here to create two new cells. So altogether, prophase, metaphase, anaphase, and telophase form the four phases of mitosis. And prophase you can think of as pro meaning the beginning, meta you can think of as middle, and ana I always think of a meaning apart, and telo means distance, like a telephone is when you phone somebody who's at a distance from you. I believe the word telephone comes from telephony, which is sending sound over a distance. So that's how I remember telo. So you need to know the order of that, P-M-A-T. Now, if we go back to our cell cycle, what we were just talking about, prophase, metaphase, anaphase, and telophase, make up the phases of mitosis. This particular diagram leaves out cytokinesis, but cytokinesis would be right here at the end. So PMAT are the stages of mitosis. If we add cytokinesis to the mix here, we get the entire cell division process, which happens from about 11 o'clock to about 1 o'clock on this diagram. And if we add in interphase with cell division, we end up with the entire cell cycle. Hope that helped, and thanks for listening.